This video goes through the information that you will need to put on your Peach note card. It's biotechnology. So something I would be familiar with is what are stem cells. So these are just cells that are undifferentiated. Scientists, doctors can use these for stem cell therapy. They can turn these into any cell that they need to study on or to help a patient. So a stem cell can be turned into muscle tissue, nerve cells, whatever cells they need, they can turn a stem cell into that. Something else important it says on this screen here is that differentiated cells can turn on and off different genes with their needs. This is known as gene expression. So we have talked about how every single one of your cells in your body was made through mitosis, which means they're all exactly the same. They have the exact same DNA. Even though they have the same DNA, they might look different. So if you look in that picture at the top of the screen there, all those different types of cells, they physically look very different from one another despite having the same DNA. And that's because they have a very different role in your body. So they are able, through what is known as gene expression, to turn on and off the genes that they need or don't need. So if I need a certain protein, I'll flip on that gene, I'll turn it on. If I don't need to make that protein, I can turn that gene off because it's a waste of my time. I don't need it on. So gene expression is the reason why cells can differentiate and turn into different cell types and do their different important jobs. DNA fingerprinting or gel electrophoresis is the process that we did in class here to analyze the different sizes of DNA fragments. The farther they go in the gel, the smaller they will be. If they hang out at the top of the gel where you put them in, they are very big and could not travel the fastest. So you can solve crimes. You can figure out who is the dad. That is typically what you use this for. So for this picture, we can see which suspect is matching up with DNA found at the crime scene. And it looks like suspect 2 matches the DNA sample found at crime scene 1, so suspect 2 is guilty. Recombinant DNA is a process where you take DNA from 2 different sources, you're splicing them together. You cut them with restriction enzymes, which gives them the same ends, either sticky ends or blunt ends, depending on the cut that was made. And you can stick them together to put in a desired gene into a strand of DNA you're working with. So a very good example of this is to make a glowfish. You can take the glow gene jellyfish, stick it in a regular fish. Well, now your regular fish can make the protein needed to glow. The last thing here is just the idea of genetically modified organisms, which can be abbreviated as GMOs. These are just organisms that have modified genes. So scientists oftentimes will modify organisms, typically food, to help to improve our amount of food, quality of food. It's done to improve our lives. So if we need corn that is resistant to a particular bacteria or insect, can change the genes in a crop such as corn to make it healthier so that we can make enough food to survive. This concludes everything that you need to include on your Peach note card.